Hello there. Do you or your loved ones suffer from blindness or vision loss? Has this been an ongoing and worsening problem since infancy? Well then, you may be suffering from Leber's Congenital Amaurosis, also known as LCA, which is a rare but devastating condition.
That sounds like what I suffer from.
Vision loss from LCA is caused by the loss of function of certain cells in your eye. These cells, known as the rod photoreceptor cells, give you the ability to see in dim lighting. As these cells deteriorate, it results in the degradation of other types of cells in the eye, such as the cone photoreceptor cells. These cells are responsible for your color vision. As the photoreceptor cells deteriorate, your retina and the blood vessels in your eye begin to thin. Because the retina is the part of your eye which detects light, thus allowing you to see, this initially causes weakened peripheral vision, or tunnel vision, during childhood, and the severity will increase until eventual blindness in adulthood.
That explains so much, but why do I have LCA?
Unlike other forms of childhood blindness, LCA can be detected in infants. It is usually passed down from two parents who carry the genetic defect for LCA, but may or may not be affected by it themselves. This means that they have normal vision, but could pass impaired vision on to you. This is because LCA is an autosomal recessive disease, so you need two copies of the mutated gene to be affected. However, LCA is sometimes caused by random new mutations. In either case, there are a variety of genes that, when mutated, can cause or contribute to LCA.
So this is all my parents' fault? So what can I do to fix it?
That's what I'm here to tell you about. We would like you or your loved ones who may have LCA to participate in our very safe, potentially legal, pending FDA approval clinical trial. Previous studies involving breeder dogs have shown that injecting a normal, unmutated version of the mutated gene in LCA can help to restore vision. This is done by inserting the healthy gene into a type of virus vector, which acts as a form of transportation for the healthy DNA, and is then injected into your eye and transfers the healthy DNA into your cells. With the healthy gene in your eye, your vision could be restored.
So what does all this have to do with me?
Well, in our very safe, potentially legal, pending FDA approval clinical trial, we would like to test this treatment mechanism on you. We simply need to inject some healthy DNA into your eye, potentially giving you the opportunity to get your sight back. Only a few spots are left to participate in this trial at the University of Notre Dame, so sign up now. And now, back to our regularly scheduled programming. Only a few spots are left to participate in this trial at the University of Notre Dame, so sign up now. And now, back to our regularly scheduled programming.
Hi, I'm not blind, but I saw an infomercial about your research, and I'd like to get involved.
All right, let's talk about it more inside. Welcome to the Hive Lab. There's a lot we need to go over before we can get started.
Great, I'm all ears. And eyes. Haha, get it?
Alrighty, so let's get started talking about Leber's Congenital Amaurosis. It's a condition in which rod photoreceptors start to deteriorate. So with this, it can be caused by mutations in over 14 genes, but mutations in the RPE65 gene is most common. We know this because we have conducted several tests to identify the correct genes associated with LCA. For instance, we conducted the LOD score analysis test where we identified the approximate location of the genetic defect responsible for inducing the symptoms of LCA. We also analyzed the gene expression pattern to ensure the mutated genes were expressed somewhere in the eye. We can detect physical symptoms of LCA by examining retinal images. The presence of cytoplasmic inclusions indicates that RPE cells are dying. Normally, RPE cells are responsible for the uptake of waste from rods and cones, but extra cytoplasm means that they are not endocytosing properly. So as you can see in this image of the retina, RPE cells are non-functioning, so you can see the buildup of cytoplasmic inclusions. Okay, so LCA results from malfunctioning of retinal pigmented epithelial cells, leading to failure of recycling all transretinol. This causes a defect in the phototransduction cycle. Normally, all transretinol goes back to RPE cells to be recharged, where a functional RPE65 enzyme converts all transretinol to 11-cis-retinol. 11-cis-retinol is then oxidized to 11-cis-retinal, which travels back to the outer segment to combine with opsin to form rhodopsin, resetting the phototransduction cycle. In individuals with LCA, however, the RPE65 enzyme is non-functional, so all transretinol is not recycled and the phototransduction comes to a halt. This first occurs in rod cells before taking place in cone cells. As the photoreceptor cells malfunction and blood vessels thin, vision becomes tunnel-like. However, the photoreceptors are still alive and viable, so there's hope for gene therapy. That is why we are here with the clinical trial. LCA is caused by a loss-of-function mutation, so we can add back normal function with the wild-type gene. First, the wild-type gene is identified, but then because DNA is long, mRNA is selected so that the introns are spliced out and then it becomes smaller. Second, an adeno-associated viral vector is used to transfer the gene to the specifically associated affected region of the individual. Third, the promoter is used to moderate the transcription of RNA. Alas, the function of the photoreceptor cells and RPE cells are restored. The goal of this trial is an improvement in vision.
Awesome! That sounds like a real wild type of gene therapy. Get it?
After further consideration, there's no spots in my lab. Good luck elsewhere. Thank you.